Hello, hi, Assalamu alaikum. My name is Waqas Ashraf. This presentation is recorded to the third International Brain and Biomedicine Conference 2026. My topic is genome annotation and in silico characterization of biocontrol related genes in an indigenous bacillus strain. These are my outlines. Introduction. Soil-borne fungal pathogens like Fusarium rhizoctonia and Alternaria cause 20 to 40% annual crop losses worldwide. Chemical fungicides are effective but raise environmental and resistant concerns. PGPR offer eco-friendly disease management. Bacillus species are particularly promising due to spore-forming ability and production of antifungal metabolites. Strain TD11, indigenous bacillus species TD11 isolated from the local rhizospheric soil of Dera Ismail Khan, KP, Pakistan, previously reported by Malik et al. 2022 to suppress tomato fungal pathogens via chitolytic activity. Rationale of the study. Information on the genome of an isolated strain of Bacillus from local regions remain limited. Strains are isolated from other regions or model strain often show difficulty in adapting to local soil conditions. This study examined an indigenous vegetative strain of Bacillus isolated from the local environment using computational methods and approaches for analyzing the genome. This analysis identifies genes, proteins, and nucleotide sequences that are used as biocontrol against fungi. Objectives: to assemble and annotate the draft genome of an indigenous bacillus strain with putative biocontrol potential. Number two, to identify and characterize biocontrol associated genes and BCGs using advanced in silico computational tools. Methodology. This is the pipeline. First of all, we use Windows 11 and we set up the Linux for and Windows 11. Then we retrieved our query genome and reference genome from GenBank. Then we check the quality of our genome by FastQC and trim the low reads by Trigonometric and reassemble the genome by speeds and annotate our genome for CDS and tRNAs for, by PROCA and circular visualization by proxy. Anti-smash and BCG profit identify different BCGs. Multi-gene blast compare the all genome and big fam analysis show the distance of our query genome from other bacillus genus genome. Deep CMS used for the prediction of structure and function of the novel genome, novel clades, novel nodes. CSZY database show the biocontrol enzymes. MEFT, by MEFT we use the, for alignment. Fast ANI show the average nucleotide identity. And last but not least, Mega, we built a tree by Mega. Target organism and genome retrieval. We target the indigenous bacillus strain isolated from the local environment and previously identified by Malik et al. For tomato fungal pathogens. This project was funded by Git Genome Project. The excision number for our query sequence is this. Use a single and elemental reads and FastQ file. Comparative analysis and tree building. For comparative analysis, we retrieve different bacillus strains. This is our query sequence, query genome, and these are the reference genome retrieved from NCBI. And this is the outgroup genome. These are all the name of the strains, and these are all the excision number of every particular genome. Results. Quality control and read processing. When we apply the fast QC, high quality data were obtained, approximately 2.95 million reads. Every read has 36 to 251 base pair. GC content is 34%, which is constant all over the reads. Strong score mean Q30 plus across all reads. Clean library, no adapter contamination found. Trigonometric keep 100% of the reads. This is the table and this is the graph which is created by the FastQC and Trigonometric. We reassemble our genome after the Trigonometric. We found 21 to 127 kmers. We found our genome complete from 21 to 127 chimeras, we found our genome complete. We annotate our genome by PROCA. We found 6.25 million base pair the length of our genome and 96 condex, 600, 184 CDS, 104 tRNAs, 14 rRNAs, and 1 tmRNA. This is the circular map of our genome created by proxy. We can saw that uniform CDS distribution. Biosynthetic gene cluster analysis using anti-smash identified 18 means 18 BCGs across 13 contexts, covering 9 BCGs classes, including NRPS, PKS, RIPPS, trypin, and cytopore, etc. BCG profit analysis, we found another 284 BCGs for trypin and 100 for alkaloid, and these are all the old, which is already mentioned and in anti-smash analysis. Discovery of novel BCGs, multi-gene blast analysis. We compare our query sequence, query genome with 7 reference bacillus genome. Several clusters show homology with bacillus cereus and bacillus subtilis, but these 3 clusters show no significant homology with any other reference genome, node 1, node 30, and node 38. Now we use big fam analysis and check the distance of the 3 nodes, which is pretty good. And these are all no similarity with bacillus, but some similarity with some other bacteria. Predictions, structural prediction of novel nodes using deep CMS AI tool. This is the node 1, which is similarly with polyene pyrrolidine dione derivative. Node 30, which is the similarly with staclosine C3, and node 38, which is similarly with tyrosine. Function of node 1. Mostly function of node 1 may be enzyme inhibition, membrane interruption, possibly antifungal and antibacterial activity. Likely function of node 1 as antimicrobial or enzyme inhibitor. Function prediction of node 30. Likely function of node 30 as antimicrobial peptide because the molecular weight is very high. Sulfur suggests membrane disruption, pore formation, and bactericidal activity. Function prediction of node 38. Likely function of node 38 is broad spectrum antibacterial peptide because broad spectrum antibacterial activity, disruptions of cell membrane integrity, use and topical antibiotics. This is the analysis of DBCAN3. We find different genes already published in Hammer, Diamond, and DBCAN database. These are all the biocontrol genes present in our genome, like chitinase, cellulase, glucokinase, protease, and these are the gene ID present in our genome, and this is the function of every particular gene. Fast ANI taxonomic placement. This is our query genome, and these are the reference genome. So our query genome is closely related with the Bacillus cereus, which is 98.41% similarity. And the phylogenetic tree of 16S RNA shows that Bacillus species TD11 is closely related with the Bacillus cereus. So this is the conclusion of our entire presentation. We found bacteria and rhizospheric of the Ismail Khan soil. The total length of our genome is 6.25 million base pair, and all these are information are here. This is the acknowledgement because we have less time and didn't discuss these acknowledgement and conclusion. This is the references. Thank you.